igneous and metamorphic rocks. Some of them are called garbage can minerals, like hornblende, that's calcium and sodium. Also magnesium, iron and aluminum, silica, iron, I've forgotten what OH is, but anyway. They're called garbage can minerals because of all the stuff that can have in it. So thank you, Amber.
Okay, so we're just getting up to the top here, getting caught up with Argus, and we'll assess and try to follow that ridge up.
Great, thank you.
Steve, just to confirm, that was 130. Copy. Yeah.
There's a
Brisingid.
Yeah, Brisingid. So that's the first one I think we've seen today.
Yeah. A lot of echinoderms on this dive.
This looks a little more pillowy here, so maybe this is volcanic. Just a little bit worried down below what we were seeing. Could it be a sponge? No. Well, it's sponge base, dead sponge base.
That's what I want to see when we get to the top.
These fallen sponges are probably really interesting habitat for looking at macrofauna. Oh, there's a bamboo coral to the right-hand side, a big isidella. Those are just off the screen. Yeah, I was thinking the fallen sponges, when they create these debris piles, probably have tons of little macrofauna in there that might be interesting for biologists who work on crustaceans, or polychaete worms or things like these. They probably present pretty interesting habitats for small critters that are potentially dissimilar from what inhabits these sponges when they're live.
Now these definitely look like pillows here. Bit of a relief.
It might still take us over the other face, but might not. Might flatten out.
Thanks for tuning in from around the world today. It's a great time of day, seems like, for viewers to be following along. This is our regular 8 to 12 shift with one minor alteration. We have Chris Kelly joining us as our guest scientist from the, what's your regular watch, Chris?
The next one.
The 12 to 4. Pulling double duty today, giving Beth a little bit of a break as she's been busy processing samples. It's been an exciting dive so far. We've been on the seafloor here on Euphemia Seamount for about an hour and a half, collecting rocks and biological samples. Just generally characterizing this region.
Do you have any more questions from the viewers that maybe we could answer while we're getting caught up to the ship?
Yeah, if you have questions, feel free to throw those in the message box on nautiluslive.org. There's a little green light that says, ask us your questions. Here's a geology question for both our biologists here, but
Uh-oh.
is lava that cools underwater harder than lava that cools in the air?
I don't know, Amber, did you hear that? Is lava that cools underwater harder than lava that cools in the air? Well, I think the lava that cools in water cools faster. You know, if I'm not mistaken, if the lava cools still in the lava tube, if you will, and it's not exposed to the water, it cools more slowly and actually becomes harder, perhaps. But lava that comes in contact with water cools very quickly, and I think sometimes that can make it a little more brittle. And air, you know, air isn't quite such a heat conductor as water is, so it's going to cool more slowly, I think, naturally in air. So that would be my guess. So Amber, let's see, I don't think it affects the density though. So you think that on air and in water, it could be pretty much a similar density, and it's not related to temperature, or we're waiting for wing species typing.
Yeah, and how much would the deep sea pressure impact that as well?
That's true. A lot of
That's an interesting point. I wonder if the pressure down here keeps the gases in solution, basically. Good question.
Yeah, thanks
Save
for your
that
question.
for the next watch. Tectonic turtles, right? Is that them?
what is our
12 to 4 is the tectonic turtles.
Tentacular turtles? Tectonic? I can't.
Intertentacular tectonic turtles.
Now we're naming other watches, not ourselves.
Yeah, I don't know. What is the solubility of gases in lava? I don't know. But
well, sometimes if it, and again, correct me if I'm giving bad information, Amber, but sometimes when the eruption occurs close to the surface, you can get little vesicles that are formed, which is the gas erupting from the surface of the lava, so it has a pitted appearance. And I don't think that happens as much down deep, perhaps because of the pressure. That was always my understanding.
like pumice material?
Yeah. Well, you actually see it, you know, and that we were on one of the geologist seamounts in the main Hawaiian Islands and had a geologist tell us that's how you could tell that there was an eruption pretty close to the surface. At high pressures, the gas will be dissolved in the melt, so you probably wouldn't get that vesicle formation. She also says that composition can be different, but the timing of the cooling will be different between air and water, or down here. There are a lot of variables. And she also said on land there's a lot of gas, so the basalt can create scoria, which is full of holes called vesicles. Thank you. I don't know if you wanted the job, Amber, but you're kind of our little resident geologist on this watch since Beth isn't here.
Yeah, thanks for tuning in and following along. Switching gears from geology to ichthyology. Chris, any suspicions on why we're not seeing as many fish at this particular depth on this particular location?
Well, I'm not seeing a whole lot of prey for them, you know, and there's not a whole lot of sediment. A lot of the deep water fish seem to like to dig prey out of sediment. So you have some pockets here, but probably not enough to support a nice sort of, what do they call it, a cryptic community down there that they can get at. And then you're not seeing a lot of surface prey here, like a lot of shrimp and crabs and things like that.
When you're talking about the benthic prey, are you talking
Yeah, the benthic prey that the fish would,
worms and invertebrates there in the sediment?
yeah, that the fish would, you know, would typically feed on. So like the ophidiids, the cusk eels are going to be predatory, but there's not a whole lot of things for them to eat. I haven't seen a whole lot of things. They won't eat coral or anything like sea stars. Oh, that's just my guess. The end of the dive yesterday, we had quite a lot of fish that were showing up in there because we did have more sediment. We were on the top of the geo there.
I don't know, Chris, but I'm in a little bit of an anemone mood with all these anemones. I don't know what these small pink ones are.
You want to take a look at the next one? Point
Yeah,
one out?
I mean, they never get any love. Really. Like, no one collects them.
They're hard to collect intact.
Yeah.
Is that a sea cuke or is that one of those guys?
I think it might be an anemone, yeah. I've seen them all over.
Yeah. Should we show them some love? Just by at least taking a look at them.
Take a look, sure.
A little baby Walteria to the left, too.
Oh, yeah, right there. Yeah.
Oh, the only way we're going to be able to identify them from video is to
Good, fish full in the
sample
face.
a lot and image a lot.
You think that would suck off with a slurp?
No way of knowing until we try.
You want to try? I actually, in the hit list, if you might have looked through there, I had quite a lot of anemones in there. These particularly sort of semi-transparent.
Would you like to stop here?
Yeah, let's stop
Yeah.
and try a slurp.
Sure thing.
The other option would be like to wait and see till we find a rock, but I think we can probably slurp it here.
Okay.
Well, we could sort of do both if this doesn't work.
Full wide, please.
It's gonna
All
have to be
depends on how
quicker
much
than-
adhesion
well,
they
it'll be
have
right.
on the rock.
Sorry.
Yep.
We might have some movement, but I think we're okay.
Roger. Oh, I turned down your grip force to like 3.
Okay.
So you can turn it back up to 9 to grab that puck.
Here we go, the moment of truth.
I've been corrected, Chris. Your watch's name is Kui Kuakai.
Yes, thank you. Couldn't
What is
remember the Hawaiian version. But
Okay.
it's something sponge, I think.
Sponge team.
Sponge team, yeah.
While you're doing that, Jake, I'm going to flush for you.
Andy and Canalo can really whip that off pretty quick. I'm a little slower on the...
Jar 2 is available.
Jar 2, Raj. Nice.
Actually,
Suction zeroed.
since we've seen so many, it wouldn't be a terrible idea to get 2. We can put one entirely in ethanol and then the other in...
yeah, good.
Yeah,
and one of them
formalin
in
fix.
formalin. Yeah, very good idea.
All right.
Stand by.
Come on.
Stand by, Jake.
Yeah,
Yep.
stand by,
Oh,
Jake.
sorry. I don't know if I got it.
No, we're not sucking yet.
Or not, yeah,
I thought you
I'm
were.
still having trouble
All
with the sample jars rotating here.
Yeah, I was having
Alright,
that
we're
same
on 2.
problem. You
Alright.
have to go like one direction, and then you can go the other.
Alright, you're at 50% suction now.
Alright.
70.
You might want to zoom in just to see what. Yeah.
Yeah, can you zoom in there, Jeff, just a little bit?
It closed up.
Yeah.
So.
Try it.
It's all right. Should be a bit easier. Pop them off this way.
There you go. Hopefully that's a little better for you. Need a little butter knife to pop them off. Are you still stable? Go ahead.
Kind of hoping that that botryoidal or grape-like surface would make it more difficult for them to adhere as tightly.
They're so small, though.
Yeah, they're
Pretty
so
much surface
small,
area.
but I think they're stuck pretty good.
Yeah, they look pretty stuck on. Also, they're probably hiding out in that crevice a bit.
Yeah, maybe we ought to go to plan B, which is try to find an example of one on a small rock.
Roger that.
Yeah. No, come on.
Get a little wide there, please.
It's worth a try.
Yep.
Gotta-
Okay,
Good to
full
know.
wide.
They're yeah, smaller than I thought.
Section zeroed.
A
Nice.
little bad, outsmarted by an anemone.
That's what they do.
That's what
That's what
they do.
best.
500 million years of evolution just to outsmart the humans.
Jake, when you have a moment, can you tell us why we call that the slurp?
Sure. Yeah, it's just there's a pump that basically sucks water through a long hose. So kind of just a funny little name for it is the slurp, because it just kind of slurps up the water.
Is it basically acting like a water vacuum?
Yep.
Hoover for our British friends.
A hoover. Well, we've had pretty good luck and success today with collection. That was our first foiled attempt. But gotta add a little excitement to the dive.
So back row, just considering the duration of the rest of the dive, we have over 1 kilometer to cover, and we've gotten about 200 and some odd, 250 maybe since the start. Just for you guys to keep in mind, we can go a little bit faster, or we can keep on kind of like truck on a little bit, and when we see a sample, we can stop immediately, but if we keep stopping and starting, we might not make that 1K.
Yeah, I guess we should. I mean, we're not doing too bad because the 12-hour dive, based on Steve's calculations of 100 meters per hour, we're more or less keeping up.
Okay.
Maybe a little while longer, but yeah, we could speed it up. I'm a little worried since this is kind of more of a horizontal dive moving along the summit, that speeding up may not yield particularly richer grounds, and then we'll wind up at the end of that and just have to come back or do something weird.
Yeah.
Okay, sure thing. All right, ready for you.
Roger.
Very consolidated
Bridge
substrate
nav.
here. There's no loose rocks whatsoever.
So that'll put us
A lot of those little
Lots of
on
pigmented.
it,
yeah.
I think, and then we can follow.
Okay.
It's pretty flat.
Yeah.
Yeah, we are not on the same seamount as yesterday. Yesterday we were on Tamana seamount. We are today on a seamount called Euphemia seamount.
Oh, these
It's
anemones
like
are
a chrysogorgid in there.
taunting us.
Might be an anemone on a rock. You could,
Yeah,
Oh,
Oh,
you're
yeah.
right. All right, right down there. Yep.
And in the foreground too, another one.
Yeah.
Yeah,
that
some
one has
little
a bunch
smaller
on it.
ones.
Yeah.
yeah.
Is
Yeah.
it
You're
Down
right.
to
is it possible to stop here and take a look?
I would do it on the fly, yeah.
Okay.
You think
Jake,
you
can
can
you
do it?
arm out?
The one on the left has quite a few little anemones on it.
yeah.
It's pretty flat, so I think actually it'll pass over and be all right.
I wonder if
Yeah.
that one would be it'd have two of them on there. We might be able to do that thing Steve was talking about where one goes in alcohol and one goes in formalin. If that rock is loose.
Kind of in the pit here, so
Yeah, it's kind of a little hard place to get into, I guess.
all right, Jake, go ahead and get the arm out there. No. All right, that's great there. And then we're gonna do a quick zoom.
Before we grab.
I think so, yeah.
Right.
Go ahead and push on in there a bit, please, Jeff.
So we're more interested in those little pink ones and not the orange ones, which we know the genus of already. That's a Philiactis.
So the ones at the bottom?
Yeah, I was thinking of that kind of hexagonal rock might work.
The one to the left, to the right?
It's this one right here.
Center?
Yeah.
All right.
Oh, okay, Raj.
Good.
Can come a little wide, please? All right, go ahead, Jake.
We going in the front with this one? Steve?
Yeah, we can
Or
go
do you
anywhere.
want this side?
Yeah, there's no restrictions now.
All right, you're unrestricted, Jake.
All right.
Just not starboard C or D.
Okay.
Ah, it's loose.
Nice.
Yes.
Nice. You want to take a picture of it before we stow it?
Yep.
All right.
Ready when you are.
Maybe push it out to the brow so you get a little more light. Yeah, nice.
Oh, there's
Three
three of them.
Three
Oh.
anemones.
4. Very nice.
Yeah,
Great.
the rock isn't bad either.
All right, thank you.
All right, full wide. Make sure I'm full rack back here. All right, full rack back.
I think there's actually 5. There's one underneath one of the prongs.
They may not all come up in good condition because they are on a rock that's going to roll around a little bit, so it's not bad having more than one on different surfaces.
You know, we get a lot of questions about whether these species are able to survive the pressure change, but Steve, you mentioned that they don't really have air sinuses, so the pressure is a little, can you say more about that?
Yeah, it's, you know, I would say only the fishes really are subject to pressure changes at these depths, but these invertebrates, they don't have any air-filled spaces. So
Watch the lens.
Yep.
So there's nothing to change volume with change in pressure.
I would orient or rotate a bit more so that that one doesn't open up to the lens.
Yep.
But fishes, of course, have the swim bladders, which change in volume as pressure changes.
My elbows up. Yeah. All right, there we go.
You can also just honestly just drop it and see which one it goes into as well.
Okay.
Nice.
Thank you.
Great. Yeah, thank you.
One, 3, 1.
Right.
Nice.
Much easier than slurping. That worked out well.
Yeah, great.
And come up a bit there.
Yep.
Yeah, so we're here on Euphemia Seamount today. We're exploring this region. We've been down here on the seafloor about 2 hours now. This is scheduled to be a 12-hour dive, and that includes the descent and ascent, or the launch and recovery of the vehicles. So we should be looking at having the vehicles back on deck around 8 p.m. Hawaii Standard Time. Leaves us a bit of time to explore here.
I haven't really seen any communities I would characterize as high density or high diversity, so I haven't really been inclined to take a Niskin water sample yet, but we'll see.
Right.
As we get a bit shallower, things might pop up.
Yeah.
Compared to some of our other dives, we are relatively shallow, I guess. Oh. Renata, would it be possible to see HYPACK for our viewers?
Sure. Would you like me to zoom out?
Yeah, just zoom out a little. So you can, oh, thanks. You're off SPL.
Ship move is just about complete.
Roger.
So, Pilot, I was looking at the map, refreshing my memory on that, and yeah, this is good, and now HYPACK as well. Maybe we could pick up and go a little bit faster and try to cover this sort of flat area we're on right now until we get to that steeper slope and see if we can get into richer hunting grounds.
Yeah, we can do that. We'll start with some 0.4 or 0.5 knots and go up.
Yeah, right about there. Perfect. Maybe we'll
0.4, great.
Okay. There's Metellagorgia. It's a Chrysogorgid.
Yeah, I think a 0.4 would be nice to start when we can increase speed if necessary.
We could take a quick peek at that guy.
Just waiting till we're finished this move.
We won't be able to take.
Roger that.
If you can, if you can't, that's no problem. But just before you start on faster run here, I was thinking just the one on the right would be fine.
Sure.
Thank you.
.65, and you are.
Oh, yeah. Are you pointing out the solitary hydrozoan there, or the sponge?
The sponge. I don't see the solitary hydrozoan.
Just to the right of it.
Is that? It might just be an anemone with a.
All right, go ahead and push on in there, please, a bit.
Is it? Oh yeah, it is. The solitary hydrozoan to the right side.
Stock.
Oh, I see. Yeah, so we have two things we
What
get.
are those called again?
Those are Corymorpha.
Yeah.
Which one would you guys like to look at?
So this is a sponge called Dictyocalyx. It's kind of a weird sponge, and the stalk is really, really hard, like a hard sponge. And then all of those tubes are very soft and squishy. That's great. I just wanted to see what it is. And then maybe that guy to the right there, that solitary sticking up, the hydrozoan sticking up right there. Beautiful tulip-shaped thing here. What you look at, there's two groups that are really hard to distinguish, and this one is, I'm pretty sure, is Corymorpha because I don't know what you call that sort of orange stuff right around the disc in the center. It's got to be complete. If it's not complete and has a gap in it, then it's the other thing, brachio something or other. Brachiocerianthus, that's what it is.
Is there an extra protrusion there right in the center of the disc? What is that?
Yes, there is. And that's why we were saying that Ceriantherian that we think we collected could also be a hydrozoan because these guys also have those tentacles coming out of right around the mouth.
All right, full wide, please.
That's beautiful. Thank you very much for the...
Yeah, sure thing. That was cool.
Those Dictyocalyx sponges always remind me of little PVC pipes.
Yeah, they're pretty cool sponges.
All right, we're going to go 125.
125.
Sometimes those hydrozoans have commensals living on them as well, polychaete worms. Sometimes will live within or between the tentacles. Guessing they're grazing on anything that the hydrozoan might be eating.
Things are just gorgeous animals.
Wow,
and the other one, let's see, find a good example right there. So the other one is Brachiocerianthus. The mine actually functioned for a little while. So see that little gap, showing Lindsay this little gap here. That's the difference, believe it or not. So the Corymorpha, this stuff, I've forgotten what it's called, goes all the way around complete, and there's no gap in it. And then in Brachiocerianthus, there's a gap.
It's a little break, yeah. Wow.
Who would have known?
No. Lots of observations, I guess. Are you familiar with Chris Maw?
I know him very well.
I guess he is very happy about the Sunstar collection. He commented on the Facebook group.
I figured he'd be chiming in. Someone was going to let him know if he didn't see himself. So great.
This is likely a new species.
Well,
And that, again, that's about that solaster.
yeah, I know exactly what he's talking
Okay.
about. And I actually thought he would be interested in that one. That's why we grabbed it.
Yeah, yeah. Just letting the viewers know.
Yeah.
It's exciting.
If anyone would know if it was a new species of sea star, that's the man right there, Chris Ma. There's a Walteria sponge right there to our lower left.
Haven't seen, we were talking about this on the other watch, these benthic ctenophores that are often on the branch tips.
You're
Are
right.
those called branches on the Walteria? Or
You mean the benthic? Oh, the branches,
what
the
are those
lateralia.
projections?
Lateralia is
Yeah,
what they are.
okay. Wow, that's a great word.
Yeah.
Lateralia.
Yeah, but they're also on other species, those benthic ctenophores. They're really cool, and you're right. I haven't seen any of them. We've zoomed on plenty of sponges. There's a big bamboo in the back there.
Yeah, that actually might be opportune for Scott, who's on chat. This might be one of the sparse branchers. I'll take a closer look. It has that sparse branching silhouette.
Yeah, we got something like a regadrella in front. I don't know if we're still... Nah, it's dead again. That's a dead Walteria, never mind. But this bamboo would be...
Zoom on the bamboo, is that what the consensus?
Yeah, we're going to zoom.
All right.
Yeah, we'll make our way over there. There's also a nice current
It didn't
now.
get that
Yeah,
high
so we're at
before
the
it branched, though, right? I think he was...
I think it might be variable. Yeah, but I mean, that seems about right.
And it's kind of reddish. I guess that's what you were saying.
I have, I think here there might be some...
partial there.
I printed out
It's great.
that from Scott.
I don't know. Maybe.
It's a wicked bamboo coral in the background, though, with some
With hydroids?
weird hydroids on it, yeah.
Oh yeah, you guys going full wide there?
Yeah, it could be.
I thought we would be a bit shallow for those. They seem to be in that around 2,000 meters, 1,800 to 2,000 range.
Right.
We're now 15.
Hey, we're 15, yeah. What is going on? It's a bamboo with hydroids, right? That's what you think it
Hydroids,
is?
yeah.
Yeah. Might have died because there might have been a sea star that cleaned this whole thing off. You just never know. I don't see any stars nearby. That's a polyopogon sponge behind it too. That's facing in the other direction.
All right, Jeff, go ahead and push on in there a bit. That's good. Interesting. Get a shot of the sponge on our way out. All right, full wide, please.
Yep, hydrozoan of some kind.
Does this make you excited when you see ID guides that just have a big question mark at the end?
Well, it helps us to know what we should be looking to collect. If we have a solid species ID, there's really no point in it because we already know what it is.
Yeah. A little background knowledge for our viewers, our little insight. The back row here with our scientists, there's a lot of knowledge. They're holding a lot of knowledge in their heads, but there are also a few open field guides out and some digital photo guides as well. More of those little pink anemones. So we're coming up on our final hour here. So about an hour left of our watch, although this dive will continue until probably close to 6 p.m. Hawaii Standard Time. Another 6 hours or so. And we'll be back on deck around 8 p.m. Hawaii Standard Time. This is our 10th dive in Lu'ua Ea a Hiki Ika Papaku expedition, where we've been characterizing a chain of seamounts, trying to collect samples that will help us get information on the age, the origin, as well as the geologic story over time, and kind of characterizing some of the higher density biological communities as well, mostly sponge and coral. But we've also, as you've seen today, been collecting some other phyla as well.
But just for the viewers' information, we're kind of going over a bit of a flats, which should be obvious here, but then we're going to start heading uphill on sort of a little promontory, so it's going to get steeper. And we're going to cover these flats a little bit faster just because there's not a whole lot of life on them, and hopefully we'll find more on the steeper terrain that we're going to get to in just a bit.
Yeah, Chris, Jess was noting, and you can probably see it in the water column, that once we got to the top here, you can see the current is kind of coming from what looks like coming from the southwest to the northeast.
Really?
Yeah.
Boy, that's why we can't figure out these ridges, because the current seems to be different on every ridge we...
Well, maybe, does that sound right, Jess?
Yeah, that sounds correct.
Yeah.
Yeah. The other day it was east, and then another it was northeast to southwest. And it just, maybe it's, I don't know, maybe the seamounts themselves cause some type of swirling going on, or eddies
There's
or something.
little fish.
Where's that?
Oh, at 8 o'clock, 7 o'clock. Almost out of view. There, it's gone.
Oh, okay.
Yeah, there's a little one. Yeah, I know some of our viewers have been trying to actually map the currents and keep track of it for us as we've been moving along, which appreciate. I think there's a lot of complicating factors there.
Yeah, one thing when Emma was talking about internal waves, it's like, you know, we presume that everything down here, you know, isn't really changing too much, but it could be that there's temporal changes.
Oh, this is going to be fish. Big one.
There we go.
Oh, we need to see this. Oh, well, no, it's Synaptobranchid. I was looking at the wrong end and thought it was, the head was on the left, but it isn't. But yeah, let's see it anyway. Our first fish that we had a chance to image here. It's a cutthroat eel.
That's good there. Well, he's going the right direction, so we'll chase him.
It's got to be a meter long.
High speed chase.
High speed chase here.
I'm going to add another step.
Go ahead.
Bridge nav. One more step, 100 meters, 125.
You know,
We'll
ahead,
still be
forward, please.
0.4. Thank you.
one of the keys to going further than the family Synaptobranchidae is the placement of the dorsal fin on its back. And it was very difficult to see. It's not
It was
a
kind
high
of collapsed down, wasn't it?
Yeah, and that's they're small, but it's the origin of that. And there's one particular species called Synaptobranchus brevidorsalis, and the name actually is very descriptive. It has a very short dorsal fin toward the rear end of the fish, and that's what brevidorsalis is referring to. And then a lot of the other ones, the actual dorsal fin originates further up on the body quite a bit. I couldn't really tell, so I think we're just going to leave this one as Synaptobranchus. Whoa,
maybe
here
he'll
he is
come back.
again.
Wow.
He says, do you
Oh,
want to
yeah.
see my dorsal fin?
Go ahead and
There you
fish
go.
on in a bit.
It looks pretty far back, doesn't it?
I think so. Like where it stops, yeah.
Starts right, well, it starts right behind that
Yeah.
scarring there, kind of halfway back.
Yeah, I wonder if Ken Sulak, maybe he's
He's
not
like,
on.
nope.
When they do that.
All right.
Yeah, we could,
See you later.
Very,
Consider-
very thin, isn't it?
yeah, he is. I don't think he's doing very well here. He seems, there's not a whole lot to eat. I think he's Synaptobrancos. Maybe we can go to genus and leave it at that. He's definitely not the other genus, Iliophis, that I'm most familiar with, that has white lateral line pores on it that are quite visible usually, and they tend to be skinnier eels, but this guy was skinny. His head was big, so it looked to me like he was kind of half starved or something. That's not typical of these
We
guys.
use the term eel commonly for a lot of these long, skinny fish, but are they technically true eels?
Yeah, this one is.
Cool.
But the rat tails and the cusk eels are not. They're fish. They just resemble eels.
Someone's wondering what these the little lines, wispy lines are. I believe they're little coral whips or something.
I think so. That are dead. There may be the remnants of either unbranched primnoids or unbranched isidids, or keratoisidids. That looks keratoisidid, I think. I think I saw a node.
Yeah. Agreed.
So in other words, an unbranched bamboo coral that's been stripped of all of its tissue, or an unbranched primnoid coral. Oh, big guy. It's a chimera, Hydrolagus purpurescens. And
Too fast for us.
Yeah, he's right.
Almost behind the
Argus
boat now.
view, yeah.
You can see it in Argus view over to the left of Hercules, swimming off to the left.
Yeah, they're really cool fish. That's about as active as I've ever seen one of these things.
Big pectoral fins,
Yeah.
like wings.
They're also called ghost fish, or
Looks like
chimera.
it's turning back around, like it might come back toward Hercules.
That's the guy.
Chimera. Are those, so would those be cartilaginous fish?
Yes, yes it is, exactly. It's most closely related to sharks and rays than it is to bony fishes. Well, very weird fish. I don't know much about them.
What are, so I'm looking at a close-up photo of this Chimeridae, and I'm seeing these, are these little Lorenzini here on the front? No, don't know, okay. I wish you could have seen Chris's face.
I have absolutely, yeah, deer caught in the headlights there. I have no idea. I should try to find out more about these fish. They're
Oh,
really,
look, there we go.
really
I'll ask
cool.
you again. Do you know that there's a camera right in front of you there?
What's that?
Oh, you can wave to the camera.
That's not fair. Where is it? I don't even see it. No, I'm waving.
It's right there. Right there. Right here.
Oh, okay. Yikes. Now I know. Uh-oh.
Try to ask you a good reaction-worthy question again.
It doesn't make you feel too uncomfortable. It's sort of more aimed right at you, Kalama.
I've been in the headlights 90% of the time, gang. So it's all good.
There's a little stock sponge.
Kalama, anything in particular you're hoping to see on this last 40 minutes of our time here?
whatever's clever at this point. I don't know.
Maybe more animals? That would be helpful.
Big
Whatever's
stalk
clever.
there in Argus.
Yeah.
Yep.
Certainly
Dead
like to see.
bamboo with, or no, maybe it's not dead.
Oh.
Kind of looks like Charlie Brown Christmas tree.
Actually, a bolosoma sponge in the background there that's over to the right. You can tell by just the shape.
Looks like a little one in the foreground too.
Yeah, exactly.
What was that one called? Bulbosoma?
Bolosoma is what it's referring to. It's a lollipop, a big
Lollipop.
deep water lollipop. Here's another little one here
Yeah.
as well. And the first dives I ever did to this depth, I wound up in a field of those things, and it was the yellow one. And the only thing that could come to mind is, now wait a second, what planet am I on? It was just completely bizarre.
Ship has about 10 more meters left, and Argus will swing 60.
Roger.
Maybe some nodules there.
Yeah, just a little tiny patch of them.
Yeah, a weird little pocket.
Treasure.
Pocket
that will
nodules.
make our
Yeah.
nodule fans happy.
They might want to see a few more than that. That literally is could probably fit in your pocket, that cluster. I think according to what Beth Horcott was talking about, you'd think they'd need a larger sand pocket for the water to come under the sand in order to create the nodules. I didn't know that, but she was saying that that's at least part of the process. We have water coming up.
Right, you do need
underneath.
that water flow.
Yeah, but that was such a small pocket. How does the water even get in there? Sort of strange.
Current's just right there, yeah.
I guess so.
Cool feature in Argus view.
Yeah,
Definitely take
couple
a
of
look. At,
creatures at the top.
Oh, there's a bamboo coral coming up into view. You can see it in Argus.
can we get a screenshot of the Argus view?
I was sort of thinking this is more what Scott was looking at, Steve, you know, with the longer thing before it branches, but who knows? Your picture looks a lot, that he sent you looks a lot more what we imaged before.
Jeff,
Great.
what kind of camera is Argus using?
Same one.
Both Herc and Argus have an Ikegami HL43 box camera in them.
Zeus Plus.
Yeah, they're the housing and all of it's put together by Insight Pacific, and we call it a Zeus Plus, but the actual camera is an Ikegami.
Well, what's the name of the camera?
Ikegami,
Ikegami. Okay, didn't know that. New information.
and I'm pretty sure that the glass, the lens in front of it's a Fujinon lens.
And what brand are the O-ring seals, Jeff?
They're Vutron. In my circles, you can have very, very severe discussions between Fujinon and Canon. Those are the two major lens manufacturers. So
Could we add that to the words of the day? I don't know.
sure.
I think O-rings are really what make all this possible, right?
Yeah.
Oh, yeah.
Yeah. Sometimes terrifyingly so. Don't think about it too much.
If you want your head to explode, go to the McMaster car website and look at O-rings.
Yeah.
I've done that before.
Yeah.
Plenty of times.
All right, I think Argus will have another 25 meters.
Roger. Question going back to that eel, Chris, wondering if the scarring we were seeing is just because of the nature of it living in tight crevices and scratching on rocks, or if it's possible attempted predation.
It's probably attempted predation.
Something with bigger jaws than it.
Well, oh, you mean something trying to take it,
Yeah.
eat it? I think more likely it's him like smashing into the ground really hard, trying to get its own prey where it scrapes things off.
So rock scraping.
Yeah, I've actually seen that before. There's a amberjack in Hawaii. We call it the kahala. That's the sort of the typical common name for it. But I've actually seen them going after fish, and they just smash right into the rocks in the caves trying to get their prey. They seem to be very durable.
So Chris, I think we can go back to a slower speed at this point. We have about 1 kilometer left till the end of the dive, and yeah, I think we'll be all right speed-wise.
Sounds good. And we are in a little steeper terrain now too, so that would be nice.
How much more do we have? That was 125. And I think we'll do, yes, another 100 meters at 125 should be good.
This looks pretty volcanic to me. This looks like a little vent or something.
Thanks to all our viewers for tuning in for Around the World. We have lots of viewers on this dive right now, both on YouTube as well as nautiluslive.org. Thanks for all your comments and observations. And yes, as several have noted, this is the 8 to 12 watch. We have a guest of Chris Kelly joining us from the 12 to 4 watch today, giving Beth Orcutt a little bit of a break. And we're sitting here, our Hercules is at about 1,550 meters deep on Euphemia Seamount. We've been making our way up this ridge, kind of from going in a southeast direction.
We'd like a little more life than we've seen today so far, but it's been okay.
It's been a little sparse, but we've
got some good collections. There's beautiful anemones. We got that weird Cereanthid. I think we've got at least one, if not two, good rocks.
And a sea star.
And a sea star. That's right. How could I forget the sea star? I did.
Potentially a new species.
Potentially a new species, yep. There's another one of those little polyplecopherans on the rock over there, a little white spot. Wonder if we could zoom that again if we're just waiting for a moment. This is the guy right there.
Yeah, sure thing.
Trying to figure out some of these that went to genus, but there was also some that did not.
Yeah, so polyplecopheran is a mollusk known as a chitin. 8 plates, 8 hard plates on the back with a softer mantle and a kind of suction cup soft body on the bottom. We also have a hard radula for scraping and grazing along rocks.
So the one that isn't on the genus has smooth edges to its, We need a really tight zoom if that's possible. I don't know if it is.
Talking about smooth edges on the mantle or on the
No,
plates?
on the actual plates themselves. And you know, this could be a collectible. I don't know, again, if it's easy to get it off, whether it's slurp okay. But I don't know if if we're waiting for the ship to...
Well, we have to...
We can stop the ship.
Yeah, we can hold,
Could we?
but we'll try to do it quick.
Yeah, could we try it? Because this is... See, I'll show you the difference. So there's...
Full wide.
Okay. See how they're...
The ridges on,
ridges on it like that? That's the one that's described as stenosemus. And then
We'd need to get a closer look.
I think it's this one. So it doesn't really have those ridges on there. As you can see, the edges, those striations on it.
Uh-huh.
You can see the grazing trails. Well, just above it, there were some areas where it seems to have been grazed, the sediments disturbed on the rock surface. So it's been in this area for a little bit of time.
All right, it's flushed. Pull back.
So once again, that's smooth. And then if we go back to this one, oops. All those little lines all over like this.
Jar 3 is
every one of them has got scalloping.
Yeah, it's more scalloped. I noticed that the mantle on the other one also seems to come up over the edge of the plates,
Steve,
whereas this one, the plates come over the
I don't think we used 2 in the end, did we?
Oh, we didn't use 2? Yeah, that's right, because the anemone was a no-go.
It's got striations right along
so
the edge.
2 or 3?
2 or 3, doesn't matter.
Roger.
Whichever one.
It's okay.
Oops, stuck on it. Yeah.
See how it almost looks like the mantle comes up over?
Yeah, I see what you're saying. Forms a, it sort of buries the edge of the plates.
At least in that photo. Hard to
Yeah.
Pushing it out a little bit.
Yeah.
Well, this could be a potential new species, I think. I do see a difference. Oops. Got it. Stop that. Okay. Looks more like it could be the other one.
That's a good grab.
It's hard to see. It looks like the edges are exposed. Because the imagery isn't quite as good, then they left it at this polyplecophoran.
All right, push on there a bit, please, Jeff. That's good. I had to get you a little closer, Jake. Check that.
Pinky one.
Yeah, I want to watch
Yeah, what's our hose thing?
hose.
You're still good. You still have a little bit left.
A little bit.
All right, ready?
Yep.
Going on. Good, give it a scrape. Nice, solid move.
You got him.
There you
Oh,
go.
I think you got him. Nice
It's in the
collection
hose.
job. Very
Close.
nice.
Full wide, please. Go ahead and stow this puppy.
So keep an eye on channel 3 there for that polyplecophora. Yeah, a lot of comments that they might have a very tight suction, so great job.
Hey, there he is. Phew.
All right.
Took him a little
What's the
while.
sample number there? Steve?
132.
132.
Jar 3.
And we are 1550.
All right.
All right, yeah, we'll just boogie and catch up.
So you think it's that guy. And see, we don't even have the family to him. But he is just an order of him.
Come up on the winch. There,
It's
Jake.
a guess, but
Is this the most different phyla that we have collected on a single dive?
yeah, maybe. Sea star, mollusk, coral, and then, well, an anemone, a cnidarian, but a coral anemone. Yeah, we're...
Need to get a periphera just to round it out.
Yeah, exactly. We do need a sponge to round things off.